Okay, continuing with blood. So, white blood cells, often called leukocytes. Leuko means white. So white cells. So they are really cells that are used in defense of the body, in immunity. They come from the hemocytoblasts in the red bone marrow, just like the red blood cells do. They fight infection usually in the interstitium. So in order to work, they mostly leave the bloodstream. The problem with the bloodstream is the blood transports them to the area, but would wash them away from the area just as fast. So they work in the lymphatics and in the interstitium primarily. What causes them to be made is either colony-stimulating factors or interleukins. Both of these things, both of these chemicals are as the result of the body fighting off something. So if you have a bacterial infection, you will release these chemicals that cause the red bone marrow to make a whole bunch of neutrophils, and then they go to the site and they fight that bacteria. Two broad categories. There's the granulocytes that have visible granules, so granules within them, little blobs of certain proteins that you can see when they're stained. And if it likes the stain, to like something is philus, to love. So if they're stain loving, they are philic for the stain. Now the stains that we use have got two parts to it, a red part and a blue part. The red part is called eosin, the blue part is called the base. So if the cell likes the eosin, it's an eosinophil. If the cell stains with the base, it is a basophil. And if the granules stain with both, they are neutrophils because they did both. And you can see the granules. There are other types that don't have these visible granules. The granules don't stain, and they're called the agranulocytes, which means without granules. And these are the monocytes and the lymphocytes. So the granulocytes are short-lived. They are produced to do a job, and when the job's done, they're done. So the neutrophils, they stain purple, eosinophils stain red, and the basophils stain blue. The agranulocytes stick around longer. The monocytes circulate, and they last for quite a long time. When they're in the blood, they're called monocytes. When they leave the blood, they start to be able to, they become phagocytes, and they can move on their own, like amoeba. And so we call them macrophages once they leave the bloodstream. Lymphocytes are really important. The B lymphocytes and the T lymphocytes and the natural killer cells. And so that's what we are dealing with here. The agranulocytes don't say fill, the granulocytes say fill in them. And you'll also notice that the most common neutrophils, never. Next, lymphocytes, let. Monocytes, monkeys. Eosinophils, 1 to 3%, eat. And less than 1% basophils, bananas. Never let monkeys eat bananas. So here's some photographs of the various white blood cells. So this is a neutrophil. You'll notice that it's purple granules in here. It stains mostly purple. These are all red blood cells. The nucleus also has these lobes on it, so it's called a polymorpho granulocyte. So sometimes in the blood labs they call them polys. They're very distinctive. Eosinophils, the granules all stain red, tends to have 2 lobes in the nucleus. Basophils, the granules stain blue, and again, usually 2 lobes in the nucleus. Monocytes, no granules, very large nucleus, and a fair amount of cytoplasm. Lymphocytes, very little cytoplasm, very large nucleus, no granules. So we talk about this in immunity, but neutrophils and monocytes, macrophages, become really important phagocytes. Eosinophils are allergic reactions and defend against parasites, especially multicellular parasites. Basophils are all about promoting inflammation. Lymphocytes are all about specific immunity, T cells and B cells. All of these things, when they leave the bloodstream, squeeze out of the capillaries through a process called diapedesis. Dia means through, pedesis is the same word as pedal or pedestal. It's to step through. So diapedesis looks like this. There's the white blood cell stepping through into the interstitium. We normally have different numbers of the white blood cells. We want a differential to show the percentages of the 5 different types, because the relative percentages change in certain illnesses. So if you have a parasitic infection, you are going to have more eosinophils than expected. If you have a bacterial infection, you will have more neutrophils than expected. If you have a viral infection, you will have more lymphocytes, etc. We want the differential count. Now, platelets are used to be called thrombocytes, which means clot cells, and they cause clotting, or they are involved with hemostasis. They come from cells called megakaryocytes. Mega means large, karyo means body, cytes cells, large body cells. Like these cells have a large amount of cytoplasm. The megakaryocytes are made by a hormone called thrombopoietin, right? And we normally have lots of them circulating. Now, the way they are made is, let's go back. The way they are made is kind of like the thing that you used to play with Play-Doh with. You put a blob of Play-Doh in the thing and you squeeze it out through the little holes and it comes out star-shaped or whatever. Or like a pasta machine will do the same. And what happens is these cells get so big that they start pushing against the capillaries in the bone marrow, which are sinusoidal. And as they bulge into it, the blood rushing past literally rips off fragments, and those fragments are the platelets. So we have the red blood cells as biconcave discs, transport oxygen and carbon dioxide. The granulocytes, neutrophil, eosinophil, basophil, phagocytes, kills parasites. releases heparin and histamine. The agranulocytes, monocytes, which become macrophages, large particles, lymphocytes provide immunity. Platelets control blood loss. Now plasma is the other part. We have the formed elements in the plasma. Plasma is mostly water, 92% water. It transports the nutrients and things dissolved in it, regulates fluid and electrolyte balance, maintains pH, that kind of thing. Next to the water, the next most important thing are the proteins. And there's three fundamental proteins. There's the albumins, the globulins, and the fibrinogen. Albumin is called albumin because it's the same as egg white. It's the same protein that's found in egg white. Its job is to create osmosis between the blood and the interstitium. When liquid leaves the bloodstream by hydrostatic pressure, it comes back to the bloodstream by osmotic pressure, and then albumin is made by the liver and causes that osmosis. Globulins are about a third of the proteins. Alpha, beta, and gamma globulins. Alpha and beta globulins are all about transporting lipids and the fat-soluble vitamins. The gamma globulins are the antibodies, but these are globular proteins. Fibrinogen is blood coagulation proteins. It becomes fibrin when blood is clotting. So here's the little chart of that. Dissolved in the plasma, there's a little bit of oxygen, most of it is in the red blood cell, and a fair amount of carbon dioxide. The carbon dioxide reacts with the water and becomes carbonic acid. So about 7% of the carbon dioxide is carbon dioxide. 23% goes to the red blood cells, and the last 70% is bicarbonate, which is product of carbonic acid. There's a whole bunch of nutrients. So all the amino acids, all the sugars, so when blood sugar, that kind of thing. Lipids, so the fats transport in here attached to some proteins, the transport proteins, the alpha and beta globulins. There are also some waste products, and the ones that we care most about are the nitrogenous waste products. They are the result of oxidizing proteins to make ATP. Urea and creatinine are the most important of them. There's a bunch of electrolytes, the sodium, potassium, calcium, and that, and they maintain osmotic pressure and they maintain pH. So if you look at it, this is the review of the same slide we saw at the beginning.